I will start in 3-4 minutes. Right, I'll share the slides. Right, so this is the week 5 live session where I will be discussing the videos of week 5. I'll go through the classes and then at the end, we'll go through some practice problems of week 4. So we ended the last week's live session by just briefly going into HSQC, that is heteronuclear single quantum coherence. So I'll start this class by going into more details of that. Basically, we saw different proton spectra in the last class, like COSY and TOXY, and we saw that the proton spectra can become very crowded because there are so many protons in any molecule. But HSQC is a correlation between a 13C and 1H or 15N and 1H. So the two nuclei are different, and that is why it's called heteronuclear. The other thing is that if you remember in proton spectra, we had both diagonal peaks and cross peaks. but the diagonal peaks contained the same information that was there in the 1D NMR spectra. And so it had no purpose. It was just increasing the crowdedness of the spectra. So the advantage of HSQC is that you will only get cross peaks because it is the correlation between two different nuclei. The absence of diagonal peaks in the HSQC makes it less crowded. Now, we saw earlier in one of the earlier classes that the population of 13C or 15N is low. The more prevalent nuclei is 12C or 14N, which are not NMR active. So in order to observe HSQC, normally you will need to label the proteins with 13C or 15N. Basically, you will have to grow the E. coli or other system in a medium containing these 13C or 15N, so that any of the proteins that they produce are already labeled with this nuclei that is NMR active. Now, the pulse sequence of the HSQC, basically, as all, there are two channels, the 1H and the 13C channel. The initial pulse sequence is same as the inept that we studied earlier. So you have an initial proton magnetization in the Z direction, shown by HZ. A 90-degree pulse is applied on the 1H channel, and then a 180-degree pulse is applied both on 1H and 13C channel. And finally, a 90-degree Y pulse is applied again on 1H and 13C channel. And there are two time delays, equal time delays, tau 1 between the 90-180 and 180-90. So what this entire process does is that it will transfer the magnetization was initially only on 1H. The inept sequence will transfer the magnetization to 13C. Now the magnetization is in 13C. Now during the time T1, so this entire time shown by this arrow here is T1. During this T1 time, the C magnetization is evolving. So as the T1 time goes, the C magnetization is precessing in the transverse plane up till this point, where you apply another 90-degree pulse in both 1H and 13C. And what this 90-degree pulse does is that it will transfer the magnetization back to the 1H channel. So this is kind of a reverse inept. So this last sequence is kind of a reverse inept, where in inept there was a transfer from 1H to 13C, and in this kind of reverse inept sequence there is a refocusing. That is, the magnetization is again going back to the 1H. And finally, after another time delay, so this is kind of the inept sequence backwards if you think. That is why, so in inept, if you go backwards, there are 290 degrees in both 1H and 13C. Here also there are 290 degrees in 1H and 13C. Then there are time delays of tau, then 180. Here it's labeled as tau 2, then 180. So it is kind of inept sequence in the reverse order. So finally, after two time delays of T2, the FID is produced, which is collected in the 1H channel in time T2. So this is the basic pulse sequence of the inept sequence. Now, one thing here is that you start with initial proton magnetization along Z direction or HZ. When the magnetization is transferred from proton to carbon or carbon to proton, these kind of terms come. We saw these kind of terms in last week's discussions, like 2HZ-CY means that you have some HZ magnetization and some magnetization of HZ which has transferred to CY. So these are called, these are terms which have two magnetization components. Similarly, when you have a transfer from C back to H, you will have two magnetization components. But ultimately, so now that it has transferred back, why are you not immediately collecting from we are not immediately collecting the FID from here. We don't do this. We give a time delay of 1 tau to 2 tau 2. This is because we want the magnetization. So this kind of two component magnetization terms are called antiphase. We want the magnetization to come back to in phase. That is, it will only have one term like Hx. Only then we can detect the magnetization properly, because this kind of in phase magnetization is due to single quantum coherence. If you remember in the last class, we talked about single quantum coherence, double quantum coherence, zero quantum coherence, and only single quantum coherence can be detected. So that is why this time delay is given, so that the two component magnetization terms can cancel out and you are left with only one component magnetization, also called in phase magnetization, that is detected during this time T2. Now, between the pulses applied in the 1H and 13C channels, there is no interference because there is a wide difference in frequency between 1H and 13C. If you remember the 1D spectra of 1H, it was around 0 ppm to maximum 12 or 13 ppm. Whereas in 13C, the range was much larger. It went up to 180, 190 ppm. So clearly, the frequencies are also very much different in 1H and 13C. And so you can selectively apply pulses either in the 1H channel or the 13C channel. Another thing we saw in the class where we studied inept is that what should be the value of this tau 1. We saw that tau 1 value should be 1 by 4j. And what is the j here? The j is CH. So suppose we have a CH bond. The J will be between the C and the H. So it is a one bond coupling. So tau 1, we saw when we studied inept, is 1 by 4J. In this case, J is the J of CH, that is one bond coupling, which is around 150 Hertz. And if you replace that, you will see tau 1 is roughly around 1.7 millisecond. Tau 2 can be varied, but in the simplest case, it is kept as equal to tau 1. And that is the basic procedure by which HSQC is done. Now we look at the spectra. So in HSQC, you will have one frequency F1 as 13C or 15N and the other frequency F2 as 1H. So more than the 13C and 1H, the more common HSQC you will see is between 15N and 1H. This is this is what is observed basically in proteins. So proteins, as you know, are made up of peptides. And basically, here a polypeptide is shown. You have an N-terminal NH2, each amino acid. So if you see this dotted region, each dotted region is one amino acid. So this is one amino acid, this is the second amino acid, and so on. So each amino acid basically has one NH2 group. This one is called the C alpha, one C alpha to which an R group is attached, and a CO. I'll just discard this marking right now. So in this amino acid, we will look at the amide NH. So amide bond is basically this bond, the CONH bond is the amide bond, and we will be looking at this NH. So again, NH is one bond coupling, and we will be looking at the 15N versus, so here HN refers to the amide nitrogen, which is this, amide hydrogen, which is this hydrogen I've shown here. And 15N is the amide nitrogen. So each peptide will have one NH. And so one peak will be there for each peptide. provided that the side chain or R group does not have any amide NH. So suppose there are two amino acids which have an amide NH in the side group. We will see that later. But other than those two amino acids, and proline is a special case. Proline is an amino acid where you don't have a NH in the amide, because proline is a secondary amine, you can see here. So there is no NH. If no NH is there, there is no NH correlation, and proline will not have a peak. So with the exception of proline and two other amino acids, glutamine and asparagine, which we'll see later, all other amino acids have just one NH. And so there will be one peak per residue. And here basically you can see this portion that is quite crowded is the aliphatic residues. It has been expanded, and you can see that there is some labeling on each peak. Like this peak, it's written V3. V is the one letter code for valine. So this is the valine residue in the third position. Similarly, you can see each cross peak has been labeled by the one letter code of that amino acid and the number in the sequence that it occupies. So basically, this kind of HSQC spectra between 15N and 1H is the fingerprint of the protein with respect to its primary structure or sequence. So from this, we can figure out what amino acids are there in the protein and what is the structure or sequence of the protein. Normally, what is done is HSQC is combined with either TOXI or NOSI. So in HSQC, what happens? The magnetization will transfer from 1H to 15N or 13C and back to 1H. So we just saw this. Magnetization is in 1H, it transfers to 13C or 15N, and then it goes back again to 1H. So suppose you have two amino acids, valine is shown here and glycine is shown here. Both of these will show one, like glycine here, NH2 is shown, but when it is in an amide bond, it will be NH and then CO. So there will be just one NH and so on. Imagine these two amino acids, valine and glycine, in a polypeptide. So you will have two peaks, one due to valine shown in red and one due to glycine shown in purple here in the HSQC spectra. Now, if you combine HSQC with TOXI, so initially the magnetization transfer in HSQC was from 1H to 15N and back to 1H. Now, instead of collecting the data from this 1H, if you insert a TOXI pulse program, then the magnetization will transfer from the amide H to the, so this is the amide H. It will transfer from the amide H, it will go to C alpha, C beta, C gamma, and so on, which is basically what happens in TOXI. So then you will get more peaks. Here, again, if you imagine 15N in the y-axis and 1H in the x-axis, the initial two peaks are the ones which are the HSQC peaks between the amide NH and 15N. Now, the after HSQC, when you have the TOXI sequence, so this red peak is the amide NH, amide hydrogen of valine. From the amide hydrogen of valine, it will transfer to the C alpha. From C alpha, it will go to C beta. From C beta, it will go to both the C gammas. So you can see this transfer is amide H to C alpha to C beta to both the C gammas. So you are observing the TOXI peaks as well. Similarly, in glycine, from the amide H, you will go to both the C alphas. You're going to both the C alphas. So this kind of combined 2D experiment, it will give you an example, it will give you an idea of what the amino acid is. because not only can you identify the amide cross peak, but you can also identify the other cross peaks that are there in the amino acid side chain. So if you combine toxi with HSQC, this kind of magnetization transfer will occur within the same residue. But if you combine toxi with nosy, you can have correlations between two residues. We know nosy is a through space effect. So toxi cross peaks you can only observe within the same residue. But nosy cross peaks can be observed between two or more residues if they are close enough to each other within 5 angstrom. So you can either combine HSQC with toxi or nosy to get more data. And the pulse sequence is simply the pulse sequence of, so we initially have the pulse sequence of HSQC. You can see this. So initially is the inept portion, where in the inept portion there is a magnetization transfer to the nitrogen. Then you have a time T1 where there is evolution of the magnetization in nitrogen. Then magnetization is transferred back to the hydrogen, and you give a time delay of 2 tau 2. Now, if this was HSQC, the FID would have been collected here. But because you are combining toxi, after this you include the pulse sequence of toxi. And you collect the FID after the pulse sequence of toxi. So basically, by adding pulse sequences like this, you can get more information about the molecule. We will look into the very basics of multidimensional NMR right now. So this is 2D NMR shown here. The lower plane that is here is 1H, 1H. This can be toxi or cozy. It is correlation between the amide hydrogen on one side and some other hydrogen, which can be alpha, beta, gamma, anything. And the peak that is above is the two-dimensional HSQC. So you have 1H on one axis and 15N on the other axis. Now we know in HSQC, the number of peaks is equal to the number of amino acids. Provided that we are ignoring right now proline and asparagine and glutamine will have more than one peak. So minimum, if we are ignoring proline, asparagine and glutamine right now, 3 peaks in HSQC means at least 3 amino acids. So these 3 amino acids have the same HN chemical shift. So if you draw a normal like this, you can see they are coming at the same value of HN. So they have the same HN chemical shift, and in 1H, the spectra is very crowded. So you can actually combine these two 2D spectras to form a 3-dimensional spectra. In the 3-dimensional spectra, you have, so this plane forms the plane here, which is 1H versus the amide nitrogen, and the 15N of the other 2D spectra forms the third axis or the Z axis. So two 2D spectra combine is kind of giving you a 3D spectra. Now what the 3D spectra will do is that it will separate out the 3 different amino acids. So at one, so the third axis I already told you is 15N. So the 15N chemical shift of the 3 amino acids will be different. Say for amino acid 1 it is here, for amino acid 2 it is here, and for amino acid 3 it is here. So it is separating out, and within the same amino acid you will be able to see the toxi or nosy. Here we have taken toxi, so you will be able to see the toxic correlations. That is your alpha, beta, gamma, all these correlations you will now be able to see much more clearly. So the spectra here that was very much crowded has now been separated and spread out, so you can more easily distinguish the peaks. So that is an advantage of increasing the dimensionality of the NMR. That is, if you have a large protein, you will have so many cross peaks in 2D NMR that you will not be able to distinguish. But if you add a third axis, you will spread out. So you have the same number of peaks, 4, 5, 6, 7, 8, 9, and 10. You can see the 10th is kind of behind here. And here also you have 4, 5, 6, 7, 8, 9, 10. You have 10 peaks. They are just spread out in 3 dimensions. So it is easier to distinguish. Now, suppose even in 3 dimension, it's too crowded. Then for further separation, you can visualize a 4th dimension. Now, 4 dimension, obviously, you can't visualize it on a slide. But if you think about it, so what is the third dimension doing? If you imagine this 2D here, what happened here is that all the amino acids work together. But when you have 3D, each amino acid separates out into 3 different planes. So this is amino acid 1, this is amino acid 2, this is amino acid 3. So different amino acids come in different planes. Now what 4D will do, that the atoms of each, so suppose this is alpha, this is beta, this is gamma, this is delta of amino acid 2. What this 4D will do, it will separate out the alpha, beta, gamma, delta also. So just imagine this is alpha, beta, gamma, delta, so on. So 4D will further separate out the alpha, beta, gamma, delta. So if there are ambiguities, even after 3D NMR, the 4D NMR can be used for further separation. Now, so for 3D experiments, you need 3 frequency axes, F1, F2, and F3. So correspondingly, you will need 3 time axes, T1, T2, and T3. And if you are giving 3 times, so T3 is the time in which the FID is collected. And if there are 2 other time axes, there will be 2 mixing times, M1 and M2. So this is the very basic schematics of a 3-dimensional NMR experiment, where you have a certain pulse sequence and you have the evolution time T1. Then you have a mixing of magnetization. Then you have evolution of time T2. Then you have another mixing of magnetization. And finally, you have the detection time T3, in which the FID is collected. Because there are 3 time axes, there will be 3 corresponding frequency axes on Fourier transform, and the 3 frequency axes will form a 3D spectrum. So this is the very basics. We'll go into more detail a bit later. So experimental 3D spectra will basically look like this. It will be a 3-dimensional space, which is like a cube over here. So the red dots that you see, they are the 3D spectra. Now, if you make projections of the dots on the different planes, so this red dot, I am projecting it on the this plane, which is the 13C 15N plane. So if I project the dots like this on the plane, then you will get a 2-dimensional. You can see that this plane is forming a 2D spectra. And what 2D spectra is this? This is the 13C 15N 2D spectra. Then if you project the dots on this plane, that projection will give you another 2D spectra, which is the 1H 13C 2D spectra. And finally, you can also project the dots on this plane, and that will give you another 2D spectra, which is the 15N 1H 2D spectra. So projections on the plane will give you 2D spectra. And then similarly from the plane, if you project onto, so suppose I have a dot on the plane and I'm projecting it on the axis, then you will get this projection that I've shown. this will give you the 15N chemical shift, and this will give you the 13C chemical shift. So from the projection, if you further project onto an individual axis, you will get the chemical shift in that particular axis. And this is basically how 3D NMR spectra works. Now we look more into the details of HSQC. So we saw that in proteins, the HSQC that is most used is the 1H15N HSQC. And each protein, so there will be one peak per residue, as I already discussed. So you can see that each peak here is labeled by a single letter code. So if you see this peak, it's G. G is glycine. Glycine is G. So this is glycine and it is 58 in the sequence. If you look at this peak, it is L. L is leucine and it is 74 in the sequence. So like this, there will be one peak per residue except those amino acids which have an amide group in their side chain. So I told that you will talk about these later. So right now we'll talk about this. So basically, this is a asparagine and this is your C alpha. Now the alpha NH, there will be only one NH because it will be connected to the previous or the next amino acid by an amide bond. So there is one NH which is attached to the alpha carbon, and the side chain or R group is a CH2CONH2. So there is another NH2 group. These are amino acids with a side chain amide group. Then is glutamine is also there, which is same as asparagine with one extra carbon. So these will have two peaks per residue. One peak due to the main chain NH that is attached to the alpha carbon, which is this NH attached to the alpha carbon, and another is due to the side chain NH. So if you look at the peaks here, you can see just so it is not labeled by a number here, but you can clearly see this is one peak, this is another peak. So it has two peaks. So this is either an asparagine or a glutamine. Again, you have one peak here and one peak here. And it is labeled as a Q. Q is not visible here, but it's Q. And Q is glutamine, which you know should have two peaks. So these two amino acids will have two peaks in the 1H15 and HSQC. So basically, the advantage of this HSQC is that suppose I am drawing one line like this along the y-axis. So whatever, so the this is basically this value is the chemical shift of the amide proton. So all these proteins have the exact same amide proton chemical shift. So if you were taking the 1D NMR spectra of this protein, they will all show the same 1H amide chemical shift. So how do you distinguish them? You distinguish them based on the 15N chemical shift. So this is having a different 15N, this is having a different 15N, likewise. Same for the x-axis. Suppose I draw a line like this. So all the amino acids on this line have the same 15N chemical shift, and you are able to distinguish them because their amide H has different chemical shift. So that is the advantage of the 2D spectra, is that it is spreading out the peaks compared to a 1D spectra. Now we'll look at, so we were talking about how HSQC can be combined with TOXI or it can combine with NOSI. So here is the example of such a combined 2D spectra. The first one is HSQC combined with TOXI and the second one is HSQC combined with NOSI. Of the compound strychnine, which is shown here, you can see there are so many carbons. They have been numbered 1, 2, 3, 4, so on. And there are different hydrogens with different numbers also. So if you look at the HSQC nosy spectra, you will see that the peaks will have two different colors. Two different colors basically signifies two different signs. That is, they are antiphase. So the red peaks are generally negative and the black peaks are generally the positive peaks. So if you remember, in one of the previous classes we had seen that when we have a small molecule and we do the nosy spectra of that small molecule, we had seen this kind of spectra. So small molecule, just imagine these are the diagonal peaks and these are the cross peaks. So for a small molecule NOE spectra, the diagonal peaks were positive like this and the cross peaks were negative. They are going below the plane. Strychnine is considered as a small molecule, and because this is heteronuclear, so 1H and 13C, so because it is heteronuclear, there are no diagonal peaks. So the cross peaks are negative, and these negative peaks are the ones that appeared as red. So the black peaks are the peaks due to, so we know that the nosy peaks of strychnine, so in the HSQC nosy, we will observe only cross peaks because the two axes have different frequencies, different atoms. And so if we are observing only cross peaks in the nosy, all the nosy peaks will be negative. And so all these red peaks that you see are due to nosy, and the black peaks that you see are the HSQC peaks, which are positive. So this is an advantage of the HS. So in HSQC toxy, all the peaks will have the same phase, that is positive. So you will not be able to distinguish that which is due to HSQC, which is due to toxy. But the advantage of HSQC nosy is that you can easily distinguish because of the difference in phase. And these NMR software that are there from Bluecard or Geol, they are designed in such a way that if the peaks are in different phase, the cross peaks will appear in different colors. So in this case, you can understand the black peaks are the peaks which are only due to HSQC. So if we start from that, you can see, let us first look at the HSQC toxy. So there is one black peak here, which is due to the HSQC, which is the same black peak which appears here, because the HSQC will be same in both the cases. The only difference is one is toxy, one is nosy. So if you start from this amide NH, you can imagine this as alpha, beta, gamma. So from amide NH, so this peak is due to one particular amino acid because it's appearing in the HSQC. Whatever correlations this peak shows in the same line are within that same amino acid because we are doing toxy. So the correlations will be with alpha, beta, gamma within the same amino acid. Similarly, if I choose this peak, so this is the HSQC peak of another amino acid. If you go in a horizontal line, you will be able to see the toxic cross peaks. And similarly, in case of nosy, you will be able to understand the proximity with other amino acids within the polypeptide chain. So this is how you will get information from this kind of combined 2D spectrum. Now we'll go into something completely new, which is called triple resonance experiments. Just give me 2 minutes. Right. So far what we were seeing in the 2D experiments, I'll just escape this. So if you see this, We saw in the 2D experiments that there were two channels, the 1H channel and the 13C channel. In the triple resonance experiments, what happens is that you have three channels, 1H, 13C, and 15N. So in the HSQC, you saw that there were specific pulses applied in 1H, specific pulses applied in 13C. And in triple resonance, pulses are also applied along 15N. But ultimately, the final data or FID is always collected along the 1H. So we will look at some examples of triple resonance experiment. One is HNCA. Here what happens, basically whatever is shown by these blue circles, those are the nuclei that are observed in the 3D experiment. So amide N is blue circled. So this N that you're observing is the amide N. Similarly, amide H, which is also written as, amide H is often written as HN. So you're observing HN, this is the HN. And then you are also observing C alpha, that is the 13C. So this is actually C alpha and C alpha. So the arrows that are shown are the pathways of transfer of magnetization, and they are also called as out and back pathways. So in HNCA, what happens? The initial pulse is applied to the amide hydrogen. It can be transferred to the amide nitrogen, and from the amide nitrogen, it can be transferred to the C alpha. Now note that the C alpha of the same residue, so this shorter arrow is with the C alpha of the same residue, and the longer arrow is with the C alpha of the previous residue. So both C alpha are getting detected. This happens in the HNCA. In HNCOCA, what happens is that, so this transfer from the amide nitrogen and also the magnetization, so you can see the arrows are double-headed arrows. That means the pulse begins from the amide hydrogen, it gets transfers to the N and the C alpha, but then it comes back to the amide hydrogen because you have to detect this FID on the hydrogen. So just remember that this is the reversible pathway. In HNCOCA, it is a similar thing, but here the transfer occurs via the carbonyl group. Now the transfer is occurring via the carbonyl, but the carbonyl is not getting detected. In this case, the carbonyl is said to be a mediator. So you can see here, first magnetization, initial magnetization is at amide hydrogen, then it goes to the amide nitrogen, and then using the carbonyl as a mediator, it transfers to the C alpha. Now, because this experiment is using the carbonyl as a mediator, it cannot transfer. This transfer is not possible because there is no carbonyl here. This particular example requires a carbonyl group to enable the transfer, and so it only transfers to the C alpha of the previous residue. It cannot transfer to the C alpha of the same residue. And this is called HNCOCA. So you can imagine this as an extension of the HNCA, but using the carbonyl as a mediator. So its carbonyl is CO is written because it is on the pathway, but it is not detected. You can see it's not marked by a blue circle. Then you have the HNCO experiment, which is a much shorter experiment where the transfer starts from the amide hydrogen, goes to the amide nitrogen, and then the carbonyl carbon of the previous residue. And then it again goes back to the amide hydrogen where it is detected. So it is called HNCO. Another experiment is the HNCACO. This case is similar to HNCO. By that I mean that it goes from, but again here, it is kind of, you can see here the transfer is directly from N to carbonyl. But in HNCaCO, if you remember, I said that anything written in the bracket means that the magnetization passes through that atom, but that atom is not detected. So you start from HN, so you start from the amide hydrogen, go to the amide nitrogen, and then you use the C alpha as a mediator. So it is jumping to C alpha, um, but the C alpha is in bracket, so it is not detecting the C alpha. From C alpha, it is going to the carbonyl CO. Similarly, it is going to the C alpha on the other side, and from that it is going to the next carbonyl CO. So what you are detecting here is the amide hydrogen, the amide nitrogen, and the carbonyl carbon of the previous residue and the same residue. So it is kind of like HNCA. In HNCA, you are going from amide hydrogen to nitrogen and then jumping to the 2C alpha on either side of the amide NH. In this case, that part is similar. You're going from hydrogen to nitrogen and then jumping to the 2C alpha. The extension is that it's not staying on C alpha, you're not detecting C alpha. Instead, it is going to the carbonyl next to the C alpha and you're detecting the carbonyl carbon. Another experiment is so in all these experiments, you are starting from the amide hydrogen. In fact, these two experiments also, if you look at these two, so in the experiments so far, you are only traveling within the main chain. You are either detecting C alpha or NH or CO. You did not, the magnetization was not transferred into the side chain. In these two experiments, magnetization is also transferred into the side chain. So you can detect the side chain as well. This experiment is HN. So you're starting from the amide hydrogen to the amide nitrogen. Then Ca is in bracket. So magnetization is going on Ca, but it is not detected. So from the nitrogen, you can see magnetization is going to the C. Let me just label these. This is alpha, this is beta. Similarly, this is alpha, this is beta. So magnetization is going from amide hydrogen to amide nitrogen. Then it is going to the C alpha on both sides, but it is not being detected. C alpha is in bracket. From C alpha, it is transferred to C beta, and it is the C beta that is being detected, shown by the red circles here. So you're detecting amide hydrogen, amide nitrogen, and the C beta. C alpha is simply kind of a mediator through which the magnetization is passing, but it is not being detected. Similarly, you have another experiment where you're starting from the amide hydrogen to the amide nitrogen. In this case, the magnetization is passing through the CO, then the C alpha. So it is passing through CO and then again I'll label this. This is alpha, this is beta. So magnetization is coming from the amide hydrogen to the amide nitrogen. Then it is passing through the CO, passing through the C alpha, and finally coming on C beta, which is being detected. So here in HNCACB, you were detecting the C beta on both sides. In this HNCOCACB, you are detecting C beta on only one side. Why is that? Because here the magnetization needs to pass through CO, then C alpha. But on this side, you don't have a CO. So magnetization can only transfer to the previous residue. That is why it's HNCOCA, then CB. So these two experiments, side chains, can be detected. So far the 6 experiments that we have seen, this 1, 2, 3, 4, 5, and 6, you are all starting the initial pulse from the amide hydrogen. In these two last experiments, you are starting from H beta and H alpha. So this experiment, that is CbCaCONH, you are starting from H beta. You can see, again, I'll label this. This is beta. This is alpha. Again, this is. So you're starting from H beta, the magnetization is coming to C beta. And because H beta and H alpha are close in frequency, when you are exciting H beta, H alpha is also getting excited. So initial magnetization is partly on H alpha and partly on H beta. From the H alpha and H beta, it is coming to the C alpha. From H beta, it is coming to C beta, and then C alpha. From H alpha, it's coming to C alpha. Then from C alpha is being, so you are detecting C beta, you're also detecting C alpha. It's shown in blue here. Then from C alpha, it is passing through the carbonyl, but it is not detecting the carbonyl. You can see the carbonyl is in brackets here. And from carbonyl, it is going to the amide N, which is detected, and then to the amide H, which is detected. So because here you're ending at amide H, the arrows are not reversible because we always try to end at the hydrogen. So here the arrows are only in single direction compared to the other experiments. In the other experiments, you were starting from the amide H, so the arrows needed to be reversible so that you could come back to the amide H and detect the final FID over there. And the final experiment is CbCaNH, where again you are starting from H beta. It's coming from H beta to C beta, which is detected, then to C alpha, which is detected. And as I said, if you're exciting H beta, H alpha also gets excited. So from H alpha also, it is coming to C alpha, which is detected. Now from C alpha, it will go to the amide NH on both sides. And here there is nothing in bracket. So there is no mediator. So the magnetization will directly jump to the amide N and then the amide H. Similarly to the amide N on the other side and then the amide H. So here you're detecting the C beta, C alpha, amide N and amide H. The difference between these two experiments is that this experiment uses CO as a mediator. So you can only transfer the magnetization within the, you can only transfer the magnetization to the NH of the next residue. You cannot access the NH of the same residue because there is no carbonyl over here. But in the final case, you can access the NH of both the same residue and the next residue because here no carbonyl is needed as mediator to transfer the magnetization. So like this, there are different experiments and for the 3D NMR, and these are known as triple resonance experiments. So the transfer that is occurring in these triple resonance experiments, so all these transfers we have shown by different arrows, is basically through the J coupling values. And the J coupling values are shown in hertz here. So mostly these are single bond coupling. This C beta, C alpha is single bond, C alpha CO is single bond. There's only one two bond coupling here shown by this arrow, which is from C alpha to NH. Now, the advantage of these one bond and two bond correlation is that they are always the same irrespective of the amino acid or conformation of the protein. So there is no chance of missing a correlation. You will always find these correlations in your 3D NMR. And unlike nosy, so in nosy, there is a chance that if two hydrogens are more than 5 angstrom away from each other, that correlation will not be present. But here it is always one bond and only one two bond correlation. And these are close enough that you will always see these correlations. So more the number of correlations you can see, more you can walk through the polypeptide and find out what, identify that particular amino acid and also identify the amino acids before it or after it. And that will help you understand the sequence. Now, these experiments are useful for large proteins which get too crowded in the nosy spectrum. If you see the 2D spectra that I talked about, if it is getting too crowded in the 2D spectra, we do the 3D NMR so that the crowdedness can reduce and you can easily distinguish between the peaks. That is what this point is saying, is that in large proteins, the 2D spectra is very crowded. So then the 3D spectra is done because the cross peaks will get dispersed in the third dimension, and then you can easily identify the cross peaks. Now we look at some of the pulse sequences of this triple resonance experiments. So we have HNCA, but there is a modified version of HNCA called CTHNCA, or CT stands for constant time. So we will briefly look into the pulse sequence of this. We don't need to go too much in the detail. So the excitation of HNCA starts from the amide H, also written as HN. That is what is shown here. You start the magnetization from the amide H, and there is an inept sequence. Inept sequence, we know, will transfer the magnetization to the less sensitive nuclei, that is 15N. That is this transfer from the amide hydrogen to the nitrogen. Then there will be evolution of magnetization of this amide nitrogen for a time period, or total time period shown by this green arrow, which is T. So the amide nitrogen magnetization is evolving for a total time period of capital T, and this T is constant. So because this T is constant, it is called constant time HNC. And it is, so how this time period is defined is that you have T1 by 2, small t1 by 2, capital T by 2, and then capital T by 2 minus small t1 by 2. So the small t1 by 2 will cancel out, and you will have the entire time period T as constant. So T is constant, but you can vary the values of small t1 in such a way that you will observe a chemical shift evolution. So like that, you will obtain the first time axis. Then after this constant time of capital T is over, then there is another inept kind of experiment which transfers the magnetization from 15N to 13C alpha, which is this transfer, 15N to 13C alpha. You can see it has transferred here. Then there is a time period of small t2, and during this time period of T2, the magnetization or chemical shift evolution of the C alpha is happening, because now the magnetization is on the C alpha, and that is what is evolving. So this gives you the second time axis or the second frequency axis. After that, 90-degree pulses are applied so that it's kind of like the reverse inept we talked about. The magnetization then flows back to the 15N, which is the reverse arrow. From C alpha, it is going back to the 15N, and you give it another constant time of capital T, and then another reverse inept is there where the magnetization flows back to the amide hydrogen. You can see this reversible arrow, the magnetization flowing back to the amide hydrogen. And then you allow a certain time period of 2 tau so that the magnetization can be refocused. If you remember, in HSQC, we talked about that when the magnetization comes back to the 1H, it cannot be immediately detected because there can be antiphase terms. A certain pulse sequence and time needs to be given to it so that the antiphase magnetization can be refocused back to in-phase magnetization. And then this in-phase magnetization is collected in the time T3. So you have 3 incremental times, T1, T2, and T3. And these times, T1 is on the nitrogen, T2 is on the C alpha, and T3 is on the amide hydrogen. So these 3 incremental times correspond to the 3 frequency axes F1, F2, and F3 that is forming. And that is what is giving rise to your 3D spectrum. So this is kind of the in this particular experiment, there is some role of the 13C carbonyl also, but we will not discuss about that in detail because we are not going that much into the mathematics of this. So now we will look at the CTHNCA. So we talked about the pulse sequence. Now we will talk about how the CTHNCA looks like. So here a 3D cube is shown, and you have uh, 4 sets of peaks in 4 different colors. Each color basically represents, so 4 colors correspond to 4 amino acids. Each amino acid has a strong, so if we look at this green colored amino acid, we have a strong peak due to interactions within the same residue, and you have a weak peak due to interactions with the residue before or after it. So if you see the HNCA, the transfer of magnetization from the N to this C alpha is transfer of magnetization within the same residue. It is also called a self peak, and this is strong because of smaller distance. But the transfer of magnetization from the N to the C alpha of the previous residue is called a sequential peak because it gives an idea about the sequence of the polypeptide. And this peak is the weaker peak because there is a longer distance between the nitrogen and the C alpha. So each amino acid will have this strong peak, that is the self peak, and the weak peak or the sequential peak for the I minus first, that is the previous residue. Now suppose we take a cross section at a particular value of 15N. So you can see 15N is this axis. We are taking a cross section like this. When we take this cross section, which is shown here, the stronger peak will be the peak, self peak, which we are denoting as I, and the weaker peak will be the previous peak, which we are denoting as I minus 1. So like this, we can correlate different cross sections. So suppose we start from this green peak. We start from here. I'll just clear all this. So suppose we start from here. If we go up, we will find the sequential peak, that is the peak with the previous C alpha. Then if we go here, this will be the self peak. So from C alpha, it is supposed to go to NH. So this peak, that is the self peak of the I minus first residue. This is the I minus first residue. It is the self peak. Then if you go up, it is the cross peak of the I minus first residue. So I minus first with I minus second. Then again, if you go down, it is the self peak of the I minus 2 residue. Then if you go up, that is the cross peak, that is I minus 2 with I minus 3. Similarly, you can move through the 3D spectra and you can get the amino acid sequence from that. So this is a very useful method of not only identifying the amino acid, but also understanding the sequence. Now there is another kind. Now suppose one thing here is that one of the axis is 13C. But sometimes what happens is that the 13C's chemical shift separation is not very good. And this especially happens when you have intrinsically disordered proteins or IDPs. So in HNCA, you had one axis as the amide hydrogen, one axis as the 15N, and one axis as the 13C alpha. Now we will remove the 13C alpha axis so that we have two 15N axis and one amide hydrogen axis. This experiment is called HNN because you have one hydrogen axis and two nitrogen axis. So in this experiment, it is similar to HNCA at the beginning. You begin with the amide hydrogen. From that, magnetization is transferred to the amide nitrogen, and the amide nitrogen is detected. So whatever is shown in the bracket as T1, that forms your F1 or first frequency axis, and that is detected in the 3D spectra. So your T1 is 15N. Then from the amide N, it is transferred to the C alpha of the same residue and the C alpha of the I minus first, that is the previous residue. So C alpha of same residue, C alpha of the previous residue. From the C alpha, it goes to the amide nitrogen of, so C alpha I will go to Ni and Ni plus 1. So let me show this here. So this is C alpha I, and this will continue as NH. This is I. This is I plus 1. And this NH is I minus 1. So from C alpha I, it will go to Ni. That is this reverse peak that you can see is the C alpha I to Ni. It will also go to Ni plus 1. So it will go here. Then C alpha I minus 1 will go to Ni minus 1. So C alpha I minus 1 is this. It is going to the N of I minus 1. But it will also go to Ni, which is this. Then from the N, so now you have amide nitrogens of I plus 1 and I minus 1. And that forms your T2. So T2 or the second frequency axis is also 15N. And you are detecting three consecutive residues, I plus 1, I and I minus 1. So from the amide nitrogens of these three consecutive residues, the magnetization flows to the corresponding amide hydrogens, and that is where the FID is collected. So the amide hydrogens of I plus 1, I and I minus 1 forms T3. So that is the third frequency axis. So the third frequency axis is your amide hydrogen. So this kind of H, so first of all, it's called HNN because you have one H. Out of the three axes, one is H and the other two axes are N. It is also called a triplet filter through HSQC because you are kind of detecting, so I'll talk about that. So imagine you have the 3D spectra here. You have F1, F2 and F3 axis, and you are taking cross sections. So one is this blue cross section that is shown here, which is basically at a particular value of F2. So you're taking a cross section of the three-dimensional spectra at a particular value of F2. So basically F3 and F1 are varying. In this cross section, you will see three peaks. The blue peak is basically a self peak, so transferred within the same amino acid, and the red peaks are the cross peaks, or sequential, sorry, not cross peaks, sequential peaks, that is transferred to the previous and the next amino acid. And in this kind of a cross section, you will observe three peaks, I minus 1 and I plus 1, and the signs of the peak, whatever is the sign of the peak I, the sign of the other two peaks will be opposite. That is why this is called a triplet filter through HSQC, because if you take different cross sections, you will get different sets of 3 consecutive amino acids. So like that, you can identify different sets of 3 consecutive amino acids, and that will eventually, stitching up all that will give you an idea of the entire polypeptide sequence. So it is called triplet filter because you're filtering out the triplets, which is the 3 consecutive residues. If you take another cross section shown by red here, which is a cross section at a particular value of F1, so F2 and F3 are varying. In this case, again, you will see one self peak of the I residue and 2 cross peaks of the I-1 and I+1 residue. And again, not cross peak, sequential peak. And again, the signs of the sequential peak will be opposite to the sign of the self peak. So like that, you are able to identify the sequence. Now we know that if proline is there, there will be no peak. Proline will have no amide hydrogen. So that particular cross peak will be absent in case of proline. So suppose you have a triplet. Normally, if you have 3 amino acids, you will observe 3 peaks. But out of the 3 amino acids, if one is proline or P, you will only observe 2 peaks. Similarly, if you have 2 prolines, you will only observe 1 peak. This is the general case where you have 3 different amino acids, and the self peak, X with X is the self peak. So here we are basically considering the middle residue. So X with X, the middle residue is the one for which we are observing. That is the ith residue. So X with X is the self peak that has positive sign, and X with the I minus first or I plus first residue will have opposite signs. This we have already seen. This is the general case. Now, if one of the residues is glycine, then we have this sign pattern that you observed of the self peak. Let us consider the self peak to have positive sign. Then the sequential peaks will have negative sign. But whenever you have glycine, this sign pattern is changed. So imagine we have uh, X, G, Z. So in this case, what happens? You can see the self peak is shown by the square. So the glycine self peak shown by the square has negative sign because it is, so we are considering the square to be self peak, and if it is black, that means it is positive, and if it is white, it is negative. So the general case we saw is that the self peak is positive and the sequential peak is negative. But in glycine, if you have XGZ, then glycine, the self peak is negative, and in the cross peak, one is positive, one is negative. Similarly, if you have glycine in the first position or glycine in the third position, or 2 glycines, the sign combinations are very unique for glycine compared to, so this general case that you have shown here is valid for all amino acids except glycine. In glycine, the sign combinations are very unique, which are shown here. And of course, if you have proline, you will have one less peak because proline does not have any amide hydrogen. So what is the purpose of this unique sign combination of glycine is that they will help you, they will act as a checkpoint in the sequence. So suppose you are gathering different triplets, so different sets of triplets, and in one of the sets of triplets, the sign combination is different compared to what it is in all the others. That means in that triplet, you have a glycine. Then you can check that which specific sign combination it matches with, and you will be able to predict whether the glycine is in the middle or in the first or in the third position. And this will kind of act as a checkpoint to help you understand where you are in the polypeptide sequence. So the unique nature of glycine helps you to assign the polypeptide sequence more rapidly. Now, this is a slight modification on the HNN experiment. So I'll talk about this also. So again, you're starting from the amide hydrogen and going to the amide nitrogen of the same residue. That is your T1 axis. So T1 axis is amide nitrogen, that is 15N. Then you are going to the C prime of the preceding residue. So you are going to the here. C prime is basically the carbonyl carbon of the previous residue. From here, you are going to the C alpha of the preceding residue, which is here. From C alpha of the preceding residue, you can go to the N amide of the same residue that you are in, or you can jump to the N of the next residue. So this is already shown. You can jump here. This forms T2 or your second frequency axis, which is also a 15N. And from that corresponding amide N, you can go to the corresponding amide hydrogens. So you can come here and you can come here. So you can come to the amide hydrogens of the I-1 and I residue, and that is where the FID is collected. And so that's your final time axis, or T3, which is the amide hydrogen. So this is another kind of HNN spectra, but here you're using the carbonyl as a kind of mediator. So here what happens, here if you look, in the previous case, you had I plus 1, I minus 1. So there was a triplet here. But here you only have I and I minus 1. So what happens is that now if you take the cross section, so imagine first taking this red cross section. In this red cross section, you're choosing a particular value of F1, and F2 and F3 is varying. So you will have two peaks due to I and I minus 1, as you see here. Now, the interesting thing here is that if you take a horizontal cross section shown by this purple plane, where you have fixed the value of F2 and you're varying F1 and F3, then also you will get two cross peaks, but you will get due to I and I plus 1. So how is that done? Basically, here you're starting with I. So you're getting I minus 1 and I. But if you started with I plus 1, then you would have gotten I and I plus 1. That is basically what you're observing here. You're observing I and I plus 1 here. So how this is helping is that it is helping you understand the direction in which the polypeptide chain is going. In the previous case, you know that out of these two red peaks, one is I plus 1, one is I minus 1. But which one is I plus 1, which one is I minus 1, you cannot distinguish. But in this case, two different cross sections, one cross section will have I minus 1, the other cross section will have I plus 1. So you can clearly distinguish which is the residue before and which is the residue after. So you can derive the direction in which the polypeptide chain is going. Again, here, if you have proline, one peak will be absent. If you have two prolines, two peaks will be absent because proline does not have an amide hydrogen. And if you have glycine, the peak sign combinations are very different. Like in a normal non-gly system, the two peaks are of different signs. But if you have glycine, the two peaks are of the same sign. Similarly, if you have so imagine as I said, imagine the black to be positive. If you have glycine in the middle, both peaks are positive. If you have glycine in the first position, both peaks are negative, and so on. So again, glycine is a very special case, and the sign combinations in glycine are totally different compared to the general case. So by observing the unique sign combinations in glycine, you can immediately understand when a residue has a glycine, and that will kind of act as a checkpoint when you are deriving the sequence of the polypeptide. So now here kind of example is shown about how this is done. So you have both the HNN and the HNCN. So the polypeptide sequence is shown here going. So V5 is valine at 5th position, K6 is lysine at 6th position, and so on. So you have the polypeptide chain going in this direction, and how the movement is done. So basically, you can see each of these cross sections that are shown in rectangles here are the cross sections shown in rectangles here. We know that in an HNCN experiment, so in an HNCN experiment in general, you will have two peaks, one positive and one negative. The self peak is positive, cross peak is negative. So you can, you can see in valine, just follow the arrow. In valine, you have two peaks. The black peak or the self peak is positive, cross peak is negative. So from one cross peak, so cross peak means interaction of valine with the peak of lysine. So from one, so this is the peak of lysine. So you can now move to lysine. So you get the self peak of lysine, then you can move to the cross peak of lysine. From cross peak of lysine, so like this, you can travel through the polypeptide sequence. Each amino acid will have two peaks with different signs. The self peak is positive, the cross peak is negative. Where you are getting a different thing is when you have glycine. So if you look at this red rectangle here, the three amino acids are T, G, and K. So when you have glycine in the middle position, you can see both peaks are black, that is positive. Indeed, that is what you observe. Both peaks are black, that is positive. Then you move to the next amino acid, lysine. So the sequence here is GKT. So you have glycine in the first position. When glycine is in the first position, both peaks are positive. Both peaks are negative. Indeed, you can see both peaks are red, that is negative. So this is how glycine helps you, because the moment you see this cross section, you will know it's a glycine. And by looking at the sign combination, you will even be able to predict whether that glycine is in the middle position or in the first position, and so on. Similarly, for HNN, it is a similar thing, except in HNN, the general combination is that the self peak is positive, cross peaks are negative. So each cross section will have three peaks. You can see each cross section has three peaks. And one, that is the self peak, is positive, the other two peaks are negative. So you are moving through amino acid to amino acid. The moment you arrive at glycine, the sign pattern changes. In all the other cases, you had two red and one black peak. In glycine, you get, uh, sorry, uh, in glycine, the order you automatically changes, because in all other cases, first of all, glycine is, you can see here, it is corresponding to this case where you have two negatives and one positive, which is this case where you have glycine in the middle. Similarly, if you have glycine, if you look at this green rectangle, the two amino acids surrounding the green rectangle are GKT. So you have glycine in the beginning. In this case, the sign pattern will be exactly opposite compared so to what it was when the so in this case you had two red and one black, and in this case you have two black and one red. So the moment you go from the red rectangle, that is TGK, to the green rectangle, that is GKT. So you're moving from glycine in the middle to glycine in the first position. The signs will just reverse. In the first case, you had 2 red and 1 black. In this case, you have 2 black and 1 red. So the moment the signs reverse like this in 2 consecutive residues, you will know that there is the presence of a glycine. So again, this is how glycine is standing out from all the other different amino acids. So now we have actually looked into so many different kinds of NMR. But the purpose of this series of lectures is to look at the applications of NMR in structural biology. So far we have only looked at NMR. Now we look at some elements of structural biology so we can understand that what are we supposed to study using this NMR. So we'll just talk about some points of the structural biology in short. So I'll just give me 2 minutes. Right. So you all know the central dogma in molecular biology. All cells have nuclei which have DNA, which is your hereditary material. And DNA is basically a polymer made of a huge number of monomeric units called the nucleotides. And the total length of DNA, if it is extended in a linear fashion, is up to 10 to the power 2 centimeter. But this is all fit within a small, tiny cell. So there is a lot of folding happening in DNA. So we need to study the structure of the different conformations of DNA and how it is folding. Now this DNA goes through a process called transcription where it is converted to RNA. And from RNA, if it goes back to DNA, that is called reverse transcription. So this process in which DNA is converted to RNA happens, and especially the RNA is called as messenger RNA. In this RNA, so RNA is basically, you can consider it as one strand of the DNA. And in that strand of the DNA, we have different nitrogenous bases. We'll talk about the bases also in some time. Now, from the RNA, the proteins can be formed. That process is called translation, where from RNA you go to the proteins. Now, there is something called a genetic code where 3 nucleotides, or more commonly called a triplet, they code for 1 amino acid. So 3 nucleotides give rise to 1 amino acid. Next 3 nucleotides give rise to another amino acid. Like this, different amino acids are coupled and they form the protein. This process being called the translation. And proteins, as you know, are responsible for all biological function within the cell. So basically, we need to understand the structure and dynamics of all these molecules, that all these biological molecules, and we will do that using NMR. But before that, as I said, we need to understand what are these biological molecules and what are the conformations they can adopt. Now, as you go from DNA to RNA to proteins, the number of molecules increase. So suppose in DNA, you have more than 30,000 genes. So what is a gene? Gene is basically that portion of a DNA that is coding for a specific protein. Not all portions of DNA code for proteins. Some portions of DNA are inactive. Some may be responsible for some other function. But there are small segments in DNA which give rise to a particular mRNA. And that is the portion we are calling genes. Now there can be more than 30,000 genes, and this we are calling as genome. So in this plane, all these dots represent genes. You can imagine there are 30,000 genes. From each gene, you get an mRNA. So you will have more than 30,000 mRNA. That is called the transcriptome, because from gene to mRNA happens through transcription. Then from mRNA to protein happens through translation, but in the the set of proteins, which we will call the proteome, the number of molecules increases a lot. So it was greater than 30,000 in the transcriptome, that is greater than 30,000 mRNA. But the moment mRNA gets translated to proteins, it becomes more than 100,000 proteins. Because different mRNA can combine, called mRNA splicing, giving rise to different proteins. Then after it is translated into protein, it can undergo different post-translational modifications. So one mRNA can give rise to more than one proteins. That is so the number of molecules in the proteome increases significantly. And from the proteome you have the metabolome. Metabolome is basically the proteins are taking part in different biological functions, and they are giving rise to different metabolites or side products from these biological functions, and all these form the metabolome, and there can be more than 40,000 of these metabolites. So there are so many molecules, and understanding the structure is important to understand or predict that how these molecules can be converted from one to the other. Now, what these up or down arrows represent is that there can be up regulation or there can be down regulation of these processes. So, conversion of gene to mRNA or mRNA to protein, or protein carrying out its biological function to give rise to metabolites. All these processes, they can be upregulated, shown by the up arrow, or downregulated, shown by the down arrow. And this upregulation or downregulation depends on different environmental factors like diet, age, lifestyle, the drugs or medicines you are taking, or disease. So many diseases, what happens, they can cause some problem in the transcription, that is the conversion of gene to mRNA. Then a wrong mRNA is formed, and that will give rise to a defect in the protein. And so that defective protein will not be able to carry out the biological function properly. And so it is called down regulation. Or even if the opposite happens, that suppose a certain protein is expected to be formed in a certain quantity, and there is upregulation, that is too much of that protein is forming. So that can also be a problem. So this is basically what happens in a biological process. Now we just said that the DNA is a polymeric molecule, and of that the monomer is called a nucleotide. So we will look at the structure of the nucleotide. The nucleotide basically has a pentose or a 5-membered ring sugar, and it also has a phosphate group, and it has a nitrogenous base. So this entire unit is called a nucleotide. Here you can see it is written out in more details. So from this dotted line to this next dotted line, if we consider it the numbered N, so the nucleotide numbered N. So up to this O is one nucleotide. This O will form a phosphodiester bond with the phosphate group of the next nucleotide, which is the N+1 nucleotide. And this phosphate of the N nucleotide will form a phosphodiester bond with the O of the previous nucleotide, that is the N-1 nucleotide. So the phosphodiester bond, that is bond between phosphate and oxygen, is basically what is connecting the different nucleotide units. And the sequence DNA chain runs from 5' to 3' direction. Why is it called like this? Because this, if you just consider within the N nucleotide, this atom O is labeled as 5' and this atom O is labeled as 3'. So that is why it's said to run from 5' to 3' direction. So as the DNA sequence is running from 5' to 3' direction, it is the DNA sequence that carries all the information, because it is from this sequence that you get the proteins and then the different activities are carried out. So Again, we can look more into the structure of this five-membered sugar, which is also called the deoxyribose sugar. If you look at the deoxyribose sugar, you can see that the five-membered ring has 4 carbons and 1 oxygen. And to C4 prime, there is a CH2OH group, and to the C3 prime, there is a OH group. These two OH groups, the 3' OH and the 5' OH, are connected to phosphates. The 5' OH is connected to the previous nucleotide, and the 3' OH is connected to the next nucleotide. And in C1, there is a hydrogen. This hydrogen is replaced by the nitrogenous base so you can see the deoxyribose sugar is connected to a nitrogenous base. This happens through this hydrogen is replaced by that nitrogenous base. And you can have, so this is for DNA. In DNA, this C2 prime will have 2 hydrogen atoms, whereas in RNA, there is 1 hydrogen and 1 oxygen. Now, nitrogenous bases can be of different types. You can have 6-membered nitrogenous bases, cytosine or thymine, and thymine in RNA is replaced by uracil. Or you can have fused 6 and 5-membered nitrogenous bases, which are called purine. The 6-membered are the pyrimidine. So purines are fixed 6 and 5-membered nitrogenous bases, and they can be adenine or guanine. The primary structure of DNA is basically like this. So there is each nucleotide has a deoxyribose sugar, a nitrogenous base, and a phosphate group. It is connected to the deoxyribose sugar of the next residue, which also has a nitrogenous base, and so on. So these sugars are connected by a phosphate group, and this linkage is called a phosphodiester linkage. And this sugar and the phosphate forms the DNA backbone. Now, 2 such strands come together through hydrogen bonding to form your double helical DNA strand. And this hydrogen bonding is very specific. Adenine can only combine with thymine, and this happens through 2 hydrogen bonds. And guanine can combine with cytosine, and this happens through 3 hydrogen bonds. So the DNA backbone is formed of the deoxyribose sugar and the phosphate group, and the 2 strands of DNA are held together by this kind of hydrogen bonds. Now, within the sugar itself, the sugar or deoxyribose 5-membered ring can adopt different conformations. So we know that dihedral angle is basically the angle between 4 atoms. So suppose there are 4 atoms here, X, C1, C2 and Y. The angle, if you imagine looking at these 4 atoms through the C1. So C1 is in front, C2 is at the back, and X and Y protrude out. This angle between X and Y is called the dihedral angle. It can also be known as the angle between 2 planes. So X, C2 and C1 forms one plane, and Y, C2 and C1 forms one plane, and X, C1 and C2 forms another plane. The angle between these planes is called the dihedral angle. Similarly, in this 5-membered ring, you have different dihedral angles, nu0, nu1, nu2, nu3, nu4. And these dihedral angles have been defined by, so each dihedral angle is represented by 4 atoms. So nu0 you can see is C4, O4 prime, C1 prime, C2 prime. So on, there are 5 dihedral angles that represent this 5-membered ring. But these dihedral angles are not independent because it is a closed structure. So if you fix one dihedral angle, the others will automatically get fixed. So this is a formula for calculating the different dihedral angles of the deoxyribose sugar ring. You have nu I, so I can be replaced by 0 to 4, is tau m. Tau m is the maximum distortion of an atom from the plane of the ring. Normally, this 5-membered ring exists in such a way that 3 atoms are in plane of the ring. 1 atom is above the plane, 1 atom is below the plane. So you can see the direction, you can see the representation here. So in this case, C1 prime, C1 prime, C4 prime, and O4 prime are in the plane. That is, so just this is C1 prime, O is shown here, and this is C4 prime. These three are in the plane, and 2 prime is above the plane, 3 prime is below the plane. So 2 prime and 3 prime are distorted from the plane of the atom, and the maximum distortion is referred to as tau n. Cos P. P is basically called the pseudo rotation angle. So this dihedral angle rotation that is happening from 0 to 360 degree, or you can imagine 0 to 180, 0 to minus 180, that is the rotation, and you can imagine it. So that is called the pseudo rotation angle, or P, plus I minus 2 into delta, where delta is fixed at 140. So using this, you can, for different values of P, you can find different values of nu I. Now, the orientation of the 5-membered ring, the main orientations can be S or N. S basically stands for south, where you have the 2 prime above the plane and 3 prime below the plane. Now, if it is above the plane, we call it endo, and if it is below the plane, we call it exo. So 2 prime endo, 3 prime exo is called south or S conformations. And if it is the opposite, that is 2 prime is exo, 3 prime is endo, that is the north or N conformations. And within the S conformations or within the N conformations, the extent to which the 2 prime is above and 3 prime is below can vary. So you can see in this structure, more or less, they are equally above and below. But in this structure, 2 prime is largely above, 3 prime is closer to the plane. And in this structure, it is opposite. So like that, you can have different kinds of S and different kinds of N conformations. You can also have different conformations in the, so this was the different conformations of the 5-membered deoxyribose sugar ring. You can also have different conformations of the DNA itself. The most common conformation of DNA which we see is called bDNA, and it has basically the rise in the DNA per base pair is 3.4 angstrom. And in one turn of the DNA, you have 10 base pairs. So in one turn of the DNA, the total rise will be 3.4 into 10, that is 34 angstroms. And another feature of the bDNA is that you have 2 kinds of grooves. You can see this is a large groove, also called a major groove, and you can have a small groove, also called a minor groove. Another kind of DNA, which is mostly RNA is found in this conformation, it is called aDNA. Compared to bDNA, it has a much broader structure. And the other thing is that the base pairs are very much tilted compared to the helical axis. So if you imagine this blue line as the helix axis, you can see that the base pairs are tilted by a lot compared to bDNA. And as I said, the other difference is that bDNA is a much narrower and longer structure compared to aDNA. The third kind of DNA, also called zDNA, is comparatively rare compared to A or B DNA. It is not a proper helical structure. You can see A and B are clearly helical, but zDNA is kind of like a zigzag structure. And it is also the only DNA molecule which has left-handed helical structure. Both A and B DNA, both the helices in the double helical structure are right-handed helices, but in zDNA it is left-handed helix. And the other thing is that in A A and B DNA, the monomeric unit is a nucleotide, a single nucleotide. But in Z DNA, the monomeric unit or the repeating unit is a dinucleotide. Because C and so the thing about Z DNA is that it is rich in CG. And C has a different conformation, G has a different conformation. That is why the monomeric unit is CG together. Whereas in the case of A or B DNA, the monomeric unit is a single nucleotide, that is a single nitrogenous base. So A, B, and Z DNA can be distinguished by the different torsional angles. If you look at this structure, which I showed a few slides ago, different torsional angles are defined. You can see each torsion angle is shown by this kind of arrow and a term. So suppose we look at delta. Delta is the torsional angle between C5 prime, C4 prime, C3 prime, and O3 prime. So these four atoms, 1, 2, 3, 4. The torsion angle between these four atoms is called delta. And because C4 prime and C3 prime are part of the deoxyribose sugar ring, delta is representing the sugar ring. Similarly, you have chi. Chi is represented between chi is this angle of rotation from the C1 prime to the N of the nitrogenous base. So chi is defining the orientation of the nitrogenous base with respect to the sugar ring. Like that, you have different torsional angles for the different kinds of DNA. You don't have to remember all these in detail. You can just remember the basic thing. That is, different torsional angles are there that define the different structures of DNA. And finally, even the base pairs have different orientation. So here the base pair is shown. Now, the base pairs open up in two directions. In one direction, you have a minor groove, and in the other direction, you have the major groove. If the base pair, so here you can imagine the base pair being perpendicular to the helical z-axis, or the base pair can be tilted with respect to the y-axis, or the base pair can be tilted with respect to the x-axis. Each of these have different names like coordinate frame, tip, or inclination. Or what can happen is that instead of being perfectly aligned, the two base pairs can be opened up with respect to the x-axis, with respect to the y or the z-axis. And again, these will have different names. And the third possibility is the relative orientation between two consecutive base pairs. Instead of sitting right on top of each other parallelly, they can be twisted with respect to each other. They can have certain angle with respect to each other. And if the angle is with respect to x-axis, it is called roll. If the angle is with respect to y-axis, it is called tilt. Like this, the base pair themselves can have different orientations, and the difference in orientation can be between the two bases in a base pair, or it can be between two consecutive base pairs. And so there are so many structures and conformations possible of these biological molecules. And this is basically what we will use NMR to find. Because so far crystallography or fiber diffraction data has not been able to distinguish or identify this much detail into the conformations. So I think I've basically covered all the videos of week 5. I will quickly go through some problems of week 4. I'll solve the problems of week 4. So one problem was that in a generalized 2D experiment, preparation, evolution, mixing, and detection, where is this the FID after the detection collected? So if you remember the schematic diagram of the 2D, the preparation time was the initial 90-degree pulse. The evolution time T1 was the time in which you are allowing the magnetization to transfer. the magnetization to evolve. Then you had a mixing time, which allowed transfer of magnetization from one atom to another. And finally, you had a detection time. Now, T2 is the detection time is where the FID is collected. You can see that here the FID that is shown, it is collected during this detection time. So all collection is done explicitly at the detection time T2. And T1 is basically what? The variation of T1 generates a set of experiments with varying. So there are many experiments designed in which T1 is incrementally varied, but the final data is always collected along T2. So the correct answer is option A. Then you have this question that in a multi-pulse 2D experiment, which of the terms are observable? So we will quickly go into what happens in a two-spin system. In a two-spin system, we know first of all, according to quantum mechanics, only delta m plus minus 1 is allowed. So you can go from alpha alpha to alpha beta or beta alpha, or from beta alpha you can go beta alpha and alpha beta, you can go to beta beta. So the red arrows shown are the only allowed transitions, and these are called single quantum coherence because the m is changing by only plus minus 1. This is the only kind of transition that is observable. And how these are represented? These are represented by IKX or IKY. IKZ we normally don't see because we keep the detector in the transverse plane, so it's IKX or IKY. All other kinds of transitions, like if you, now this is a single spin term, so it's only the spin K in the X direction. You can have different spin terms, so you can have terms like 2IKX ILY, that is the spin K in the X direction and the spin L in the Y direction. These terms on their own, they have no significance, but they are basically mixtures of double quantum or Z quantum coherences. By double quantum, I mean alpha alpha to beta beta directly, where m changes by 2. And by zero quantum, I mean alpha beta to beta alpha, where there is no change in m. So a combination of these terms, these double intensity terms, can be used to obtain the pure double quantum or pure Z quantum spectra. But the point is, even if you do that, these double quantum or zero quantum spectra are not observable because we can only observe single quantum spectra. So when this question is asking which term is observable, the correct answer is option A, because the only single quantum transition is option A. B and C are basically mixtures of double quantum and zero quantum coherence, and D is basically either pure double quantum or pure zero quantum coherence. And B, C, D, none of them can be observed because the only thing we can observe are single quantum coherence. So the correct answer is A. Then similar question related to this concept is that 2IKX ILY represents what? And we have already seen that this kind of double frequency term represents a mixture of double quantum and zero quantum coherence. That is what the correct answer is D. Then again, a question based on similar theory is that in a multi-pulse experiment, the selection of pathway should end in what? So when you say the selection of pathway ending means that what should you observe at the end here? So at the end, whatever signal you get, you need to be able to observe or detect the signal. So the final signal must always be a single quantum coherence because that is the only kind of signal you can observe. So the selection pathway should end in single quantum coherence or B, because it is only if the pathway ends in single quantum coherence that you can actually detect the signal. So the correct answer is B. Then you have a question that in 2D COSY spectra, cross peaks are seen where? So COSY, we know COSY is basically cross peak between J coupled spins. So here you have a system, and suppose we are observing NH. So along the diagonal, we know the diagonal is the 1D spectrum. So we will see a cross peak between NH and H alpha. So NH and H alpha. And this is a 3J coupling because there are 3 bonds. Similarly, between H alpha and H beta. H alpha and H beta. Again, it's a 3J coupling. Then H beta to the different H gammas, which are again 3J coupling. So COSY spectra basically allows you to observe cross peaks that are J coupled. So that is the correct answer is B, and you have given that as the correct answer. The second question asks that in the 2D COSY spectra, cross peaks are seen where? Within the same amino acid or between different amino acids. So again, if you look at this, you are able to see cross peaks only within the same amino acid, because you are able to see NH to H alpha to H beta to H gamma. So all this entire thing is part of the same amino acid. Why you cannot see with the next amino acid? So if you look at the number of bonds, 1, 2, 3, 4. With the next amino acid, the number of bonds is 4. And 4 bond coupling is too small. That is why you are not able to observe with the next amino acid or the previous amino acid. So the purpose of COSY is to only observe cross peaks within the same amino acid. That is option A. So option A is the correct answer. Then you have this question of the fine structure of COSY. So in COSY, it is asking that the diagonal peak have what kind of components. So if you remember, we studied this in the previous class. So here COSY spectra is seen. You basically have a 2-spin system where both of the spins give rise to doublet. HA is split by HX and HX is split by HA. That is what is shown here. Now, so if that is the case, then each peak will have 4 components, because you have 4 components in the x-axis, 4 components in the y-axis. So each peak is made up of 4 components. And when you looked at the phases of these components, so this first peak gives rise to antiphase because they are in the opposite direction, but absorptive components. The second peak, which is a diagonal peak, gives rise to in phase because down up, then again down up. So in phase, and it is a dispersive signal. Similarly, if you look at this part of the diagonal, so this part of the diagonal is essentially the last 2. So if you look at this part of the this is also a diagonal peak. Diagonal peak is again giving rise to in phase because down up, in phase, dispersive signal. So both the diagonal peaks, so this is one diagonal peak, this is another diagonal peak, are giving rise to in phase dispersive signals. In phase dispersive. So the COSY spectra fine structure of diagonal peak will have in phase dispersive signal. The correct answer is option C. Now it is asking that in a linear 3-spin system AMX, in the cross peak between A and M, what is active coupling, what is passive coupling? So in a linear system here, AMX, suppose we are considering, we know in AMX, A will give rise to a doublet, M will give rise to a doublet of doublet, so 4 peaks, and X will give rise to a doublet. So if we are looking at the cross peak between A and M will have 4 peaks, doublet of doublet, and A will have a doublet. So direct coupling A and M is your active coupling, and all other couplings, whether it is MX or something else, is your passive coupling. So AM is active coupling, which is your option C. AM is active coupling. So the correct answer is option C. Similarly, you have a similar question for a triangular AMX system, but here it is asking that each cross peak has how many components. In this case, if you remember, in this case, if you remember, we studied this in the previous class. In a triangular system, you have doublet of doublet for all 3 spins. Now, suppose we are looking at a particular spin, the MX coupling. So X is first being split by M, and this is your active coupling. So it will give rise to antiphase signal plus minus. Then X will be split by A, and this is your passive coupling. So plus will give rise to plus, minus will give rise to minus. In other words, X will have 4 peaks, plus, plus, minus, minus. That is what you have written here, plus, plus, minus, minus. Similarly, M will also have 4 peaks, plus, plus, minus, minus, plus, plus, minus, minus. So X has 4 peaks, M has 4 peaks. So 4 into 4, there will be total of 16 components in each cross peak. That is what this question is asking. Each cross peak will have how many components? The answer is 16 components, that is D. And I think one more question is there, is that in CT-COSY experiment, splitting is removed along what axis? So the constant time COSY, what it was doing, normally this is the normal COSY, where both the axes, so this axis also has delta plus J, and the y-axis also has delta plus J. CT-COSY, what it will do is that in omega 2, delta plus J is retained. But in omega 1, it becomes only delta, J is removed. So the coupling data, or J, is removed along the omega 1 axis. That is what is happening. The splitting data is removed along the omega 1, that is the F1 axis, that is A. So that is all the things to discuss. And basically now I've covered the week 5 videos and I've discussed the questions of week 4. And as always, I'll upload the recording and you can ask me your doubts. You can just send them to me in the chat. Okay, I think I'll stop now.